these lectures by introducing you to concept. You understand? Simple concept in cancer. The whole lecture is about different concepts. These are the learning objective. These are the lectures which I'm going to go through with you. And this is the first concept. First concept is, what is the difference between a normal cell and a cancer cell? Is there any difference? You know? Is there any difference? Normal cell divide, cancer cell divide. You understand? Normal cell divide when there is a need for it, for the cell. Cancer cell divide. There is need or there is? Division and the growth of normal cell is very tightly controlled. Controlled by so many factors. Controlled to maintain tissue number. When a cell comes to a cell, خلاص, doesn't stop. When a cell contact another cell, stop dividing. Cancer cells, the opposite. Doesn't know this rule. That's why it becomes a clump, a tumor. Normal cell control cell cycle, DNA replication, cell division, entering of the cell. كله هذا ايش؟ Control. يعني it does not do it unless it's being told. Do it. Cancer cell هذا كله ما موجود. You understand? All that is not in there in cancer cell. Cell stop dividing when they dismantle. هذا ما يعرف. Continue. Need, no need, anything. It continue. It is in continuous division. While the normal cell, only when it is needed, so it could stay for a whole week, a whole month, not dividing, a whole year, not dividing. I don't know, every 24 hours. And it is طبعا faster. You understand? So that's the difference between them. وإلا anything else, يعني أشكالهم, what they contain is the same. Metabolism, same. طبعا faster metabolism. Understand? Faster metabolism. It's the same. But that's the difference. Rapid and controlled growth. One cell. Cancer is clonal. Clonal means يعني جاي من خلية واحدة. When you say somebody had cancer, do you know how to start it? It started with one cell. Only. And the whole cancer comes from the progeny of that cell. مش يعني كل التشو صار كانسر، ما فيش كلام. خلية واحدة تصير. ومن الخلية يصير كل ايش؟ كل الكم. It's clonal. So you don't have radiation and you have 1,000 or 1,000,000 cells being. No, ما فيش كلام. واحدة. واحدة كفاية. You understand? There's only one cell. So it is ايش؟ clonal. Growing mass of cancer طبعا ايش؟ This is where it's not in the normal cell. When you talk about cancer cell, most often you talk about a problem with the cell cycle. ما في مجال. Because that's the one which controls ايش؟ Cell division. So when you talk about the mutation with genes which control the cell cycle. So there is disturbance of ايش؟ Cell cycle. Cell cycle, you know the cell cycle, G1 and G0. You understand? That's what we mean, the normal cell go in G0. Cancer cell does not go in G0. You understand? Quiescent does not become quiescent. It's dividing all the time. S phase, replication of DNA, G2, and the M phase. This is the whole cell cycle. But look, there is a checkpoint. Checkpoint G1, checkpoint G2, and checkpoint M, the metaphase. There are checkpoints, meaning telling them stop, don't go any further. That's what is missing in where? In cancer. مش بكلهم, could be كلهم, but at least one. You understand? At least one checkpoint is ايش؟ non-functional. Now, regulation of cell cycle is by cyclins. You heard of that, yeah? Cyclins. How cyclins bring about their effect? Cyclins bring their effect by binding to cyclin-dependent receptors. You understand? Cyclin CDK, huh? Cyclin-dependent kinases, which are receptors, for the cyclin. So if the cyclin bind to CTK, activated, once it becomes active, this is a a kinase, phosphorylate editing, phosphorylate a protein which is usually inactive, it becomes active, that will stimulate cell cycle, will stimulate cell division. You understand? So it is by cyclin. These are the major regulator of cell cycle. That is one. So you can see cyclin dependent, which are essential for cell cycle progression. ما موجودة, there is no cell cycle, there is quiescent. Checkpoints, G1, G2, and M, these are regulate. Regulate is going, the cell going from one phase to the next phase. Growth factor is the one which affect or which influence the synthesis of cyclins. Cyclins مش موجودات 20 hours all the time. Cyclin موجودات according to the need and according to the phase of the cell cycle. Who brings that about? External stimuli. Meaning external stimuli هما growth factor. Growth factor, cell will divide. But if growth factor, cell will not divide. You understand? Because they are the one which influences the synthesis of cyclin. So these are proteins necessary for cell cycle progression, and going from one phase to the other. Tumor suppressor genes, P53 هذه أبو tumor suppressor gene كلها. P53 is ايش؟ Inhibit cancer. 50% of all cancer have a non-functional P53. What is telling you? It's telling you something else. Now we're talking about cell cycle, and we're talking about tumor suppressor gene, meaning more than one mutation probably involved in AHL in cancer. It is not just one. P53 halt the cycle if DNA is damaged under normal condition. All cells in cancer are damaged. And there is no 53. فمعنى ايش؟ You're going to keep having mutated cells. And it will not inhibit the tumor formation. And does not. Tumor P53 also stimulate apoptosis. Stimulate apoptosis to kill to destroy the damaged cells. Now there is no damage, no self-destruction, apoptosis, and therefore you have كل pushing toward progression through the cycle irrespective الخلية صاحية لو مو صاحية. And feedback mechanism, each one, you understand? Each phase stimulates the next phase. قول له جايينك يلا ابدي. You understand? So there is also a feedback stimulation of the cell cycle. So let's go back to the growth factor. 200 growth factor. What is growth factor? Growth factor is either protein or steroid. احنا نسميه بعض الأوقات hormones. Stimulate, you understand? 2 hormones. Growth factors, over 200 of them, they are protein or steroid, circulate in the blood, requires for growth and differentiation. Without them, لا growth ولا differentiation. You understand? The cell will not divide and neither will differentiate unless stimulated by growth factor. Example of growth factor: epidermal growth factor, nerve growth factor, platelet-derived growth factor, and transforming growth factor beta. These are just sample. Why sample? Because these are the most important. You understand? These are the most important, and these are the ones which we are using now in the clinic, which we use for treatment of patients. These are examples of growth factors. They have multiple function, regulation of cell cycle by affecting cell growth, differentiation, and proliferation. كلياتها هم مسؤولين عنها. Cell survival, whether the cell will survive or die, apoptosis. Cell migration, whether it migrates from one place to another place. And cell death اللي هي ايش؟ apoptosis, self-destruction. If you make too many normal, not cancer, normal cells, yeah, a lot of these cells, by the effect of growth factor, will commit suicide. Commit suicide, normal. And you bind to, you're going in a speed, and suddenly you reach the end. You should stop, but there is still some coming on the way. What's happened to them? They will commit suicide because they don't want to have cancer. The body preserves itself, by stimulating apoptosis. So the balance of a growth factor and their receptors is extremely important. Extremely important. Growth factor regulates cell cycle. Normal and cancer cells start from G, 0. Any cell cycle start from a quiescent. صحيح ولا لا؟ from G, 0. يدخل إلى G1 وبعدين يكمل. Stimulate progression through G1. So you need one in there. And we have one in there. Growth factor regulates cell death. Withdrawal of a growth factor favors self-destruction, apoptosis. Presence of a growth factor inhibit apoptosis. So حتى cell death is controlled by the growth factor. So you could have the growth factor to a receptor to a signal. I'll show you a better one later on. So I'm going there. Growth factor stimulate cyclin. Cyclin bind to CDK. Cyclin are proteins whose level fluctuate, meaning they're not always in the same level in the cell. They fluctuate according to their need. According to the need. Activation of cyclin, here CDK, regulation of progression through the cycle. So external stimuli, in this case, growth factors, and we give in a growth factor, bind to a receptor on the cell membrane. That receptor send a message, become active, send a message to start synthesizing cyclin within that cell. Cyclin bind to CDK. CDK regulate cell cycle. The cell regulate itself division. You understand? By regulating the cell cycle. Cyclin-dependent kinases are protein kinases which phosphorylate اللي شفناها. All of them are kinases, phosphorylate the target protein and make it active. Making it active. For example, CDK4/6 promote progression from G1 to S phase. You need that to do that phase changes. CDK2 function in the S phase, facilitate DNA synthesis. You understand replication? If you don't understand the major part of the cell cycle, who is DNA replication. If you don't replicate DNA, the cell will not divide. Divide على ايش؟ Replication. Mitosis is not meiosis. And CDK1 essential for the transition from G2 to the M phase. So you could see cycling. هذول فقط بعضهم. There are far more cyclins in the cell which regulate, but these are the major ones. These are the top one which regulate the thing, and that is the way which goes through the stimulating the circle. Time growth factor used in clinical medicine. Single combination. So we could use a single one and we could use a combination. This is already in practice in medicine. Latent derived growth factor, epidermal growth factor, erythropoietin, NGM-CSF, the granulocyte fat. These are the ones which are used in medicine. Erythropoietin and things. Erythropoietin is used. Where is erythropoietin produced? In the kidney. So what is stimuli? Low oxygen. صحيح ولا لا؟ Low oxygen يعني there is a problem is anemia. صحيح ولا لا؟ Because erythropoietin stimulates erythropoiesis, RBC. So when there is functional problem with the kidney and no erythropoietin is produced, what do you need to give these people? Erythropoietin. And we give them erythropoietin to treat anemia of renal disease. You understand? Because there is no erythropoietin being produced. GM-CSF, this is in bone marrow transplantation. When you ablate all the bone marrow, if somebody had cancer and you remove all the bone marrow and you put the bone marrow with the normal thing into this, you need to stimulate it. Stimulate erythropoiesis of Magali. But you use me, GM-CSF, colony stimulating factor. These are the ones which are used in medicine. You understand? This is used in medicine. So what is the role of a growth factor? In normal, growth factor proliferate and differentiation, not the presence all the time. In cancer, growth factors, all their receptors are active all the time. There is what is known as constitutive activation of the receptor. يعني the receptor is active without the presence of the growth factor. Growth factor موجود or غير موجود, they are ايش؟ active. Active due to what? Mutation. The due to ايش? Mutation in the gene. That's why they are constitutively ايش? Active. Epidermal growth factor receptor become very high in breast cancer. So it is a breast cancer marker. In triple negative TNBC, breast cancer, these do not have estrogen, do not have receptor, do not have estrogen receptor, do not express, estrogen receptor, do not express human growth factor receptor, HER2, and neither progesterone and estrogen. These are known as triple negative. This is the worst kind of breast cancer. You could have them positive and you could have them negative. This is the worst kind of cancer because you don't know what is the problem. When you have somebody positive, meaning, okay, this is the problem. But this is negative. So triple negative, you understand, will have a high epidermal growth factor. So it is a marker for TNBC, triple negative growth factor. Tumors produce growth factors such as insulin-like growth factor 1 and insulin-like growth factor 2, stimulate tumor growth, autocrine fission, meaning they stimulate the cell to divide, which produce them. So autocrine. You understand? يعني هذه مو جاية من برا، جاية من داخل الخلية نفسها. Produces these to ايش؟ Such as in breast cancer and prostate cancer. If there is a way to understand these triple negative forms of breast cancer, we've probably been able to find a cure. We don't know. We know very little about the mechanism of the mutation which bring about stimulation of cell division. There is a mutation somehow, and they're not expressed. Expressed معناه they make a protein. or they make the steroid. When there is no protein, no steroid, they are not expressed, so they are not on the cell membrane. Why? Mutation. Normal cells have limited proliferation. Meaning there is a number. We'll come to it in a minute. They proliferate and they will stop. After that, they no longer come. Post-mitotic differentiation, meaning most of them become differentiated, and differentiated cell cannot divide. صحيح ولا لا؟ قلنا stem cell فقط divide. So differentiated cell, that's why cancer mostly immature, because they are still in the stem cell phase. Once they become differentiated, fully differentiated, خلاص, there's no division, or very limited division. So programmed cell death كلها هذا absence in cancer, or disrupted in cancer. Normal cells respond to external stimuli and enter genome. Cancer cells ما فيه شيء. Does not respond to external stimuli. فالضل بي جي 1 وبعد جي 1 شو حيكون؟ اس وبعدين جي 2 وهكذا. Does not have a quiescent state phase. ما في stimuli which effective. Normal cells have a limited number of division. This is known as the high flick. Meaning high flick? Meaning there are cells, there is a number for how many they divide. And after that number of division, they can no longer divide, they die. They lose their self-replication ability. For example, embryonic stem cell and fetal cell have indefinite. They could divide to the end. بينما skin, liver cell, there is a number. Probably they can divide between 40 to 60 times. After that, the telomere is so short, no longer the cell is stable, dies. So never mind, it cannot live, it cannot divide, it dies. Neurons usually is 0. High flick number is 0. And we say it's only skin and liver, the ones which mature, fully differentiated, can divide. البقية cannot divide. So there is a number. Many cells slow down or halt proliferation when you become older. Meaning they get in the quiescent cell, stimulated, will stay there. As you get you get older. Cancer cell ignores signal to enter Q0 and continue to divide. And it will continue to divide more than normal because there is ما في high flex number. They can divide indefinitely, like a stem cell. Normal cells permanently leave the cell cycle by differentiating into a specialized cell. يعني خلاص بعد ما تروح هذه ما ندخل. Cell turning over rapidly, constantly renewed. You understand? So there is a constant renewal, such as skin, bone marrow, but there is limit. Even to that, there is a limit. When normal stem cells divide, you have two daughter cells. They have the choice. It can remain as a stem cell or it can differentiate, specialize. فيعني نص على الأقل بيكون differentiate, specialize. How about in cancer? ما في. You understand? Cells have differentiated, have reached their final specialized form. ما في هيك شيء بصير بالكانسر. Cancer cells fail to undergo terminal differentiation. That's why they are all still stem cell phase. You understand? And proliferate to form a tumor. You understand? Maybe inducing stem cell to enter post-mitotic differentiation has no capacity for proliferation in the normal. Once it are differentiated, tissue can decrease its pool of replicating cells by killing themselves. You understand? apoptosis هذا كله مو موجود في وين؟ مو موجود في الكانسر.
Doctor, if we use the growth factor to help the cell differentiate, would that be a cure?
For cancer? No. Because you don't want just differentiate, you want to stop producing them.
Yes, but if it differentiates,
مش قاعد أقول لك, it is not a matter of differentiating them. You need to produce them. No matter what you use, in production, proliferation, a lot faster than me, differentiation. And that is the problem. So this is normal self, differentiate, no divide. In the case of cancer, stem cells fail to produce one non-stem cell. يعني differentiate. كل stem cell. So they produce and they're producing a tumor. Or here, daughter cell fail to differentiate. آخر وحدة fail to differentiate. And still result in tumor. You see? So even if you differentiate them, there is a higher protection than apoptosis eliminate healthy cells during age development and eliminate damaged cells that can pose a threat to a person. So they eliminate normal cells if there are too many of them. And they eliminate damaged cells because this could lead to cancer. Mutation that compromise cells' ability to carry out apoptosis can result in a cancer does not. You understand? Inhibit apoptosis, methyl-apoptosis. This shows you a white blood cells which is undergoing apoptosis. This is a real electron microscope picture. So cancers progress from a single mutant cell. وحدة. مو ملايين which that people think. وحدة خلية واحدة سوي هالبلاوي كلها ايش؟ which shows you ايش؟ with me. You. خلية واحدة. You understand? Which shows you إنه it is accelerating so fast that overtakes the whole body. Single cell to a tumor, then to ايش؟ metastatic tumor all over the body. خلية واحدة. Cancer is multi-step process, meaning ايش؟ not one mutation. At least there are 4 to 6 different mutations. We will see it in a minute. Can take decades. Meaning ايش؟ meaning you might have a mutation will lead to a cancer. But it could take years. Could take years. Why? If the other mutation has not happened, I will come to that in a minute. You understand? It would take years because sometimes you need both genes to be inactivated. It would take years to happen. Or if you're very lucky, it could take only 6 months, such as in colorectal cancer. Until go on.
So it's easier.
Sorry? Can take decades for a single mutant cell to proliferate because it requires other mutations. Which one mutation? We will come to that in a minute. It requires other mutation. I'll show you in a minute. And what did you, somebody else had a hand? Yeah? I am coming to it. Single cell sustain a mutation in one of its growth factor regulating gene. واحدة. Growth factor تلخبطت. Another cell of the clone requires another mutation. Somehow, those who have a mutation, they are prone at having other mutations. Advantage of having another mutation. Tumor genesis progressed by clonal expansion. You will look at this. This is normal cell. One cell becomes mutated. Now the whole area will be taken up by by that. ليش؟ لأن that cell has a faster turnover of cell cycle. So it grows ايش؟ quickly and form ايش؟ a clump. بعدين وحدة. Only one cell of this gain a third mutation. And from that clone now is faster than ايش؟ the previous clone. We should all that become ايش now? Expanding. And so on. Until you reach the final one, which takes over ايش؟ the whole tissue, wherever that mutation happened. So additional mutation cause cancer cell to grow even faster. طبعا if you're talking about bone marrow مثلا yes, take over. ما في. طبعا okay. tissue كله become cancerous. Absolutely. It has a higher risk of having additional mutation than age than the normal cell. Now why? We have no idea. You understand? We have no idea. Some we have, but majority we don't seem to have. طبعا, I will tell you now some reasons. You with me? I'll tell you some reasons in a minute why they are at a higher risk of developing other mutations. I will tell you in a minute. Cancer cells, several distinct properties. Absence of contact, inhibition. Normal cells, when a cell comes to a normal cell, contact attached to a normal cell is stop. Cell division. هذا ما عنده شيء. When it's stirred, it does not go somewhere else because it's contacting ايش؟ another normal cell. تعرف هاي خدها خلاص تفضل معها. Cancer cell does not think of that. Metastasis. You understand? No contact inhibition at all. And that's why they go on top of each other in a clump, forming what is known as a tumor. If you look at this is how normal cells grow in a monolayer. You understand? This is how cancerous cells grow in a clump, which is a tumor. You understand? In a clump. ما في contact inhibition. This is cancerous cells, this is normal cells as you see them. Now, cancerous cells have reduced requirement for ايش؟ For growth factor, for example, which maintains ايش؟ Growth and ايش؟ Proliferation, because it is active irrespective. Growth factor موجودة, constitutive activation, resistant to growth inhibition. That means growth inhibition hasn't been completely removed. Cancer cells are resistant to inhibitory signals. Look at this. This is the normal growth cell. مع مين؟ مع growth factor. This is the normal growth cell without. This is cancer cell with a growth factor. This is cancer cell without. في شيء? You understand? Whether it is there or not there, it is still growing beyond what it is supposed to be. So, reduced requirement for metabolite or growth factor. Immortal. خالدات we say. Cancer cells are ايش؟ خالدة. As long as ايش؟ As long as given them all they what require. When become a clump, if there is no nutrient or oxygen or angiogenesis or oxygen reaching the ones in the middle, they will die. But if you can somehow take these cells and put them on a plate and grow them, they will stay forever. They will keep dividing and dividing and dividing forever. They are immortal. They are immortal. Cancer cells are immortal. They do not stop dividing after a predetermined number. الهاي فليكس اللي قلناها number. Maybe هيك شيء. They do not stop for generation. All cancer cells are not normal. in case of age, in case of DNA, there is chromosomal age, aberration. Look at it. This is a cancer cell. What do you see? Trisomy, monosomy. لا اكو شيء ما موجود. كل واحد بيهم هو ايش؟ الوحده. You understand? That's why it is difficult to treat. There is so many. Look at it. Look at 12. You know, every one of them is abnormal. طبعا ability to stay immortal هو ايش؟ Their ability to keep the telomerase long. In cancer cells, telomerase, the enzyme which keeps the telomere, is switched on. In me, it is switched on. In you, it is switched on. You understand? They divide to certain and then become such a short, become unstable, and that's how die. Like in cancer cell, the gene for telomerase is switched on. So it will keep the telomerase long. معناه ايش؟ معناه كل ما يخسر هو ايش؟ طولة. فيبقى للأبد immortal. Because immortality or the living of a cell depend على مين؟ Depend على telomerase. How long the telomere is? Telomere, if it leaves about 250 to 300 of its repeating unit, that's enough to cause instability to the chromosome and lead to a breakdown of the DNA. If the DNA breaks down, the cell will die. RBC, huh? Maybe a DNA dies after 120 days.
The stem cells, telomerase, you can always switch on.
Switch on. And that's what makes them immortal. You understand? If you could control it, if you could control that, in me, you could keep me 18 years old. In a girl, which is 18-year-old, she could stay 18-year-old forever. If you could control the switching on of age of telomerase. Sorry? What about? Mitochondria is a different story. But if you could do that, you could live for 1,000 years. You know what? How do you think Noah lived, if you believe Noah lived 1,000 years? Believe it or don't believe it? Huh? Believe it. I think God gave him a control telomerase gene. I wish he gave it to me, I mean. So, the difference. Contact inhibition. Normal cells are present. Cancer cells, absent. Growth factor requirement. Very high for normal cells, very low for cancer cells. Anchorage dependent, يعني stay in the same tissue. Present in normal cell, absent, metastase. Cancer goes from one place to another. Cell cycle checkpoint. كلهم in contact, intact, meaning يعني they are still working. بينما ايش؟ absent in cancer cell. Karyotyping profile, شفتوا. Understand? You just seen it, how many chromosomes are being duplicated. And proliferative lifespan, finite, because of telomere. بينما this, indefinite, because the telomerase is active. So you could see the difference between some normal cells and cancer cells. Normal tissues may proliferate continually, but numbers remain steady because the one which produce are equal to the one which are dying. You understand? Which are destructive. If too many cells are produced, the rate of apoptosis goes up. In cancer, dysregulation of growth. Dysregulation, completely high turnover. And also fail to commit apoptosis. فالاثنين مش موجودات. Normal, you have cell division equal apoptosis. إذا هذا قل وهذا زاد, you end up with with cancer. So in summary, cancer, you have selective proliferation advantage. because of too many ايش mutations. كل mutation ايش add advantage to the next clone. السل a clone خلية لها جينة يصير بها another mutation, you understand, has ايش advantage in proliferation, and this start proliferating and overtake the previous clone again from only one cell of that clone. So there is advantage. Alter the stress response. They don't need metabolite like a normal thing. You understand? They're responding high anyway. Vascularization, tumor. As the tumor becomes bigger, angiogenesis, blood vessel does not grow on the same rate. فشوف the inside is very poor vascularized, which means ايش؟ very poor supplies of ايش؟ supplies of oxygen and nutrient, and that's why they tend to ايش then to die. Invasion and metastasis. Invasion, they ran away from the immune system. They could invade the immune system. The immune system ما يعرفوا. Yes, they're normal. They're yourself. You understand? So they could evade, mean the immune system. And metastase, meaning because they could invade the immune system, they could go anywhere. You understand? You have one. For example, people say, don't have radiation of this, have radiation. No, if you have a radiation in the foot and you have some cancer cells there, they could move to where? To your brain, because they could invade the immune system and they could go metastasize to anywhere they could. Metabolic rewriting, metabolism growth factor, metabolism كله تغير تام. Especially when you have 4 chromosomes and all active, all the genes, all active in these autosomes. Imagine what you're going to have. Imagine one of the enzymes 4 مرات بدل أن يكون إيش مرتين. So there is a metabolic rewriting, immune modulation, and that's what we say. It does not, and abetting micro environment, يعني micro environment helping them, helping the cancer cells to grow and to proliferate, And this is the summary of this. So this is the introduction to to cancer. Now, do you have any question before we go to the next step?